Question 76. Question 76 asks that match the following. Match the following. In column 1, they have given A as trypsin, B as GLUT4, C as insulin, and D as collagen. Now if we match this with the column 2, in column 2 if we go on matching, trypsin is a type of enzyme. Okay, so third, it is a enzyme. Okay, and this enzyme digests protein. Okay, so digest protein. Now GLUT4 enable the glucose transport into the cell, so it matches with the 4. It enable glucose transport within cell. Now insulin matches with the, that is hormone, it is a pancreatic hormone, and D collagen is intercellular ground substance. Now in this we remember that GLUT4 is insulin dependent, means insulin trigger. In the presence of insulin, this GLUT4 transporters get found in the membrane, and which allow the transport of glucose within the cell. Now according to the matching, C is the correct answer. Next, question 77. It asks that glucose convert into lactic acid. Glucose convert into lactic acid. This process is, so here breakdown of glucose is occurring, so option C, catabolism, is the correct answer. Next is question 78. Question 78 asks that the enzyme which convert proteins into peptones. Now proteins are broken up into peptones. Peptones are nothing but soluble protein. They are the soluble protein formed in early stage of protein breakdown, and this proteins to peptone breakdown is carried out by enzyme, and the name of that enzyme is nothing but a pepsid. So B is the correct answer. Next is question 79. Question 79 asks that which of the following secondary metabolite are used as a drug. Secondary metabolite used as drug. So it is a fact-based question. We have to remember option A, that is vinblastine and curcumin are the secondary metabolite which are used as a drug. Next, question 80. Question 80 asks that which one of the following enzyme is composed of simple protein. Enzyme composed of simple protein. Now if we go with the option A is peroxidases. Peroxidases are a conjugate protein. B, phosphoglycerol are also conjugate. Option C, proteinases are also conjugate. D is given an amylase, and amylase is a simple protein, so D is the correct answer. It is a fact-based question. Question 81. Question 81 asks that as temperature changes from 3 degrees to as temperature changes from 3 degree to 45 degrees Celsius, the rate of enzyme activity will rate of enzyme activity will. So if we talk about this, if temperature increases from 3 degrees to Celsius, what happens? First, the enzyme activity increases and reaches to the optimum, and later conditions are not optimum for enzyme activity. Thus, the rate of enzyme activity decreases. Why? Because up to the temperature which can be sustained by the enzyme, up to which it will show a increase. But when the temperature is much higher beyond the capacity of enzyme, then they get decrease. So it first increase and then decrease. So B is the correct answer. Increase and then decrease. Next is question 82. Question 82 asks that cellulose is a major component of cell wall of. Cellulose is major component of cell walls of. Now we have to go here option wise. A is given as Pseudomonas. B is given as Saccharomyces. C is given as Pythium, and D is given as Xanthomonas. Now we know that in case of fungus, except Oomycetes, all the fungus have a chitinous cell wall. So if we talk about a Pseudomonas and Saccharomyces, okay, in Saccharomyces you will find a specific chitinous cell wall. In Pseudomonas you will find particularly a peptidoglycanic cell wall. Here Xanthomonas you will also find a peptidoglycanous cell wall. Pythium is a fungus which belongs to Oomycetes, and it is an exception in case of fungus that Pythium dibrinum, which belong to Oomycetes, they are known as algal fungi, and they have a cellulosic cell wall. So Pythium option C become correct answer. Next is question 83. Question 83 asks that which of the following statement about enzyme is wrong? Enzyme statement wrong. We have to select a wrong statement. Now in question 83, A is given as enzymes are denatured at high temperature. Yes, it is correct. They are beyond their capacity, they can get denatured. Enzymes are mostly proteinous, but some are lipid also. It is incorrect because enzymes are mostly protein, some RNA act as a enzyme. Then there's RNA enzyme are known as ribozymes, but they cannot be made up of lipid. So B is the particularly we can say a wrong statement. C is given as enzymes are highly specific. Yes, they are specific with the product they form. And D, enzymes require optimum pH and temperature for the maximum activity. Correct. So B is the incorrect or wrong statement with respect to question 83. Next, question 84. Question 84 asks that what is cysteine in tyrosine? Actually, there is a spelling mistake. Be remember here cysteine, the spelling is CYSTINE, and tyrosine. Actually, this both are amino acids. Yes, but cysteine is a dimer of cysteine, and these both are amino acids. So B is the correct answer. Both are amino acid. Next is question 85. Question 85 asks that sucrose is a common table sugar. It's composed of, you know that sucrose is a disaccharide and it is made up of two monosaccharide. that are D-glucose and fructose. They attached together by the aldehyde and ketone carbon and form a sucrose. So A is the correct answer. A is the correct answer for the sucrose disaccharide. Okay. Next is question 86. Question 86 asks that Jacob and Monod named those enzymes allosteric enzymes whose activity is regulated by. Now, allosteric enzymes are those enzymes which have allosteric site where effector binds, and it is extra or another site than the active site. So here with the allosteric site, the enzymes are named as allosteric because at the allosteric site the end product can bind. So option B becomes the correct answer. Next is question 87. Question 87 asks that in a DNA molecule, if cytosine is 18%, if a DNA molecule is there, you know that DNA is a double helical model. Now in DNA, if cytosine is 18%, then what is the percentage of adenine? Now you know that cytosine is 18%, then C pairs with the G, then guanine must be 18%. Now 18 plus 18, that is 36. 18 plus 18, that is 36. Now remaining out of 100 is 64%. Now 64% is divided for the A and T. So both get contributed by the 32%. So 32%, that is B become the correct answer for the adenine. Now question 88. Question 88 asks that an enzyme act by. Now we know that in each and every molecule energy of activation is found, in which a few have more while few have less. Enzyme facilitate in between two molecules, lowering their energy of activation. Okay, enzyme act or facilitate in between two molecules by lowering their energy of activation. So it lower the energy of activation and facilitate the reaction between the reactants. So A is the correct answer. Question 89. Now question 89 asks that cellulose are a polymer of. Now we know that cellulose is a polysaccharide, and this is a polysaccharide of glucose. So it is a polymer of a glucose. So B is the correct answer. Next, question 90. Question 90 asks that non-competitive inhibitor alter. Non-competitive inhibitor alter. Now option A is given as structure of enzyme. Yes, it can alter the structure of enzyme. B is given as structure of active site. It can modify structure of active site as well. Velocity of enzymatic reaction. Yes, it can modify. And D is given as all of this. So all of this is complete. So we remember non-competitive, it can modify. It cannot compete. So it is not competing with any other chemical model. It is going to act with the active site. So for this, it can modify a structure of enzyme, can be conformational change can occur. Active site will modify, and velocity of enzymatic reaction can also get modified.